Welcome everybody to an exciting edition of our webinar on emerging topics in biomolecular magnetic resonance. Both speakers of today have agreed to recording, so the lectures will be uploaded to the ICMRBS YouTube channel for those which want to listen to it a second time or dive into certain aspects. So also a reminder that you please type in your questions into the Q&A and not in the chat box, or you raise your hand and then we can unmute you. And if you're not able to answer all your questions during the Q&A, please stay on because we always have a very interesting and nice discussion, informal discussion afterwards. And there's this early career edition of the ICMRBS webinar, which I would also encourage you to participate. Without further ado, I then hand over the stage to Ed Bex, who will introduce the first speaker of today.
Thanks, Marcus. It's a particular pleasure to introduce as the first speaker today, Frank Delaglio. Many of you that have ever been to a major NMR meeting, and I know it's been a little while since the last major NMR meeting, but all of you know Frank, have met him. Frank has been a fixture of the NMR community for more than 35 years. He actually started his career, scientific career, working on X-ray scattering, but got involved in NMR spectroscopy under the tutelage of George Levy, then at Syracuse University as an undergrad student. That became a commercial company, but Frank left in early '91 to join the NIH. I had to pressure him a little bit to finish his undergraduate degree. He still needed a course in home economics, I recall, or something like that, but he eventually did get his degree. In the meantime, he developed the NMR pipe program that many of you have been using for so many years. He's done a fantastic amount of work in development of, in particular, NMR software, coming up with all kinds of creative ideas. He's got an interesting life outside of NMR spectroscopy too, but I'm not going to get into that for reasons of time. He switched to and became an independent group leader at NIST, that's the National Institute for Standards and Technology, associated with the University of Maryland about half a dozen years ago, and continues his development of innovative software. And today he's going to be talking about the applications of his work to screening and ligand studies by NMR spectroscopy. Frank, it's all yours.
Thank you so much for that amazing introduction. And of course, I'd like to thank Professor Zweckstetter and the organizers for giving us this opportunity to share the work. So, as Ad mentioned, I work at the National Institutes of Standards and Technology. We have a biomanufacturing program there with several groups. The groups are led by John Marino, and I'm going to talk about some of the work that we're doing there in biomanufacturing. The motivations for our work can be shown in this slide, which lists the sales by dollar of the top 10 drugs. And there are two interesting stories in this slide. One is, if you take a look, half of these top-selling medications are biologics. And in fact, they're biologics based on monoclonal antibodies. So one of the messages here is that these biologics are outpacing the growth of small molecule drugs, and they are vitally important, both clinically and commercially. So antibody sales are roughly $150 billion a year across the world. Now, one of the reasons why these sales numbers are so big is because biologic drugs are also very expensive. So any analytical technology that we can bring to bear that will make this class of medications more available and affordable can have a positive impact. And that's what we're trying to do in our biomanufacturing groups. Now, as we all know, in the structural biology field, protein therapeutics, in order to be safe and in order to be effective, have to be folded correctly. and they can't aggregate, because if they're not properly folded, or if they aggregate, they can cause unintended immune responses that'll either harm the efficacy of the drug or injure or even kill the patient. So this is a critical measurement need that is very distinct for the measurement needs of small molecule drug manufacturing. And what we propose is to use the idea of spectral fingerprinting to help us characterize what they call in the biopharma field the high order structure of the protein. And the idea behind it is very simple. If we have two samples and the NMR spectra are the same, we can judge that the protein structure in the two samples is also similar. And I'm going to talk today about how we can use the technique principal component analysis in a quantitative way to make determinations like this. Now, Luke Arbogast at NIST demonstrated that we can actually generate beautiful two-dimensional proton-carbon spectra of monoclonal antibodies at natural abundance. So the 2D spectrum that you see here is a natural abundance spectrum. And the reason this works is that even though a molecule like the monoclonal antibody is very big, 150 kD, the methyl groups in there are spinning around rapidly. And so even though the overall rate of protein tumbling is slow, the methyl group reorientation is very fast. And so we can generate a very beautiful spectrum that's good enough to do fingerprinting, and we can generate it in a reasonable amount of time. So that's the work of Luke Arbogast. And what I'm going to show today are methods that we can use to make decisions based on the shapes of these complicated spectra, rather than based on any detailed peak analysis or assignment. Now, in order to verify the reliability of this kind of method of spectral fingerprinting, Rob Brinson at NIST designed and organized a multi-lab study that was conducted by 26 different organizations, evenly split between academic labs, pharma and biopharma labs, and regulatory labs to test the reliability and reproducibility of this method. And in a collection of data from 39 different NMR instruments in a range of 500 megahertz to 900 megahertz, what we found is if you put standardized pulse sequences and measurement techniques in the hands of biopharma folks, that they can make these NMR measurements such that chemical shifts are reproducible to better than 6 parts per billion. So this is indeed a very reliable and robust method, and I'm going to talk about some ways to exploit it. A common exercise in this kind of spectral fingerprinting is that we have one collection of replicate spectra of one sample and another collection of replicate spectra of a second sample, and we want to decide whether or not they're the same or different. But here are examples of these two cases: 2 spectra overlaid on the left and 2 spectra overlaid on the right. I can't tell a difference very easily between these spectra, and I wonder if you can look at them and make a judgment yourself. It's rather difficult without very detailed analysis, and we'd like a way to do this that is both simpler and quantitative. So suppose we could rearrange this information like this, okay? So on the left-hand side, what we see is, while it's very difficult to tell the difference between these two spectra, if we represent the spectra in terms of a principal component analysis score plot, where we can draw confidence boundaries, we can clearly tell that these two spectra are different and these two spectra are the same, in a data presentation that doesn't require detailed analysis of the spectrum itself to make a conclusion. And so what I will talk about today are the specific details that we need to observe if we want to make a classification like this in a quantitative way. And as I mentioned, the method that we're going to use is principal component analysis, which is probably familiar to just about everyone in the audience, but you may not know the details of how it works. So we're going to talk about that in quite some detail today. So the method that we're going to use will represent each spectrum in a series as a single object in a high dimensional space, a multi-dimensional space, and the coordinates of that object that represents the spectrum will be simply all of the intensities in that spectrum. So if we have a one-dimensional spectrum with 1,000 points, we can represent that spectrum exactly as one object in a 1,000-dimensional space where the 1,000 dimensions are all of the intensities. And the same holds for 2D or 3D spectrum. If a 2D spectrum has 1,000,000 points, we can represent it exactly as one object in a 1,000,000-dimensional space. So why do we do this peculiar data representation? If you think about it, this way of representing spectra, if we take two spectra that are more or less the same, that means their intensities are the same, which means their coordinates in this high dimensional space are the same, and therefore they will cluster together in this high dimensional space. Whereas if two spectra are almost the same everywhere, except maybe there are some features that vary between the two spectra, spectra like this are going to tend to lie along lines and curves in this high dimensional space because they will have one set of coordinates in common, which are all the parts of the spectra that are similar between the two cases, and then some set of coordinates that vary continuously, which will be the parts of the spectra that are different. So this high dimensional space will have clusters and groupings according to the degree of spectral similarity and difference. And what principal component analysis does is fit this high dimensional space to a high dimensional line and use that line to orient a projection down to a smaller number of dimensions. And so we can take this 1,000,000-dimensional space and project it down to 2 or 3 dimensions so that we can view a scatter plot like the one that's seen on the screen. And the scatter plot will reveal all the different structures in this high dimensional space. So we can use this scatter plot to help make discrimination. The results of this kind of analysis can be displayed in 2 ways. One is according to what we call the principal component analysis scores, which form the scatter plot coordinates. And this vector, this principal component analysis vector, actually corresponds to what we could think of as a synthetic spectrum. So the PCA results for each component that we're going to calculate will give us a collection of scores that we can look at as a scatter plot, and it will give us a loading spectrum that we can view as actual spectral data. And I'm going to show you how we can exploit this kind of information. Now, in our case, in the biopharmaceutical area, as I mentioned, we will commonly have pairs of samples that we want to see if they are the same or if they're different, like a lot that we manufacture on one day compared to a lot that's manufactured a month later. What we see here are results for that large interlaboratory study that was designed and led by Rob Brinson. That study generated 375 different proton-carbon spectra. And Rob analyzed those by peak detection, which of course took several weeks or even months for 375 spectra. But alternatively, we can dump all 375 spectra into a principal component analysis, and without any assignments, without any peak detection, we can get a result like this, which separates all of our spectra into the 2 kinds of samples that we had. which were 2 monoclonal antibody Fab domains with 98% sequence identity. So very similar. And you can see the collection of spectra in the little animation at right. So again, this is a collection of spectra that were measured on instruments from between 500 and 900 megahertz. So the spectra are rather different in character, but we can still make a classification like this. Now, of course, most of us in structural biology are not going to be doing classifications. So what I'd like to do is show you as a tangent a couple of applications of principal component analysis to other aspects of structural biology, so your time isn't wasted by listening to the rest of the stuff that we're going to talk about. And what I'd like to convince you is that anytime you have a spectral series, it's useful to check it out with principal component analysis just to quickly see how it behaves. So for example, what we see here in overlay is a collection of 7 spectra from a ligand titration series. And we would expect that the behavior of such a ligand titration series is continuous somehow. And so if we perform a principal component analysis and look at the results with respect to concentration, we should expect to see a continuous curve. And in fact, we do. So that's a nice quick analysis that we can do without any peak detection or assignment. Another example of this is looking at a 2D relaxation series. Okay, so principal component analysis gives us these principal component spectra that show us parts of the spectra that behave similarly to each other. So if we apply this to a relaxation series, in a typical case, most of the peaks will have about the same relaxation behavior. So in a case like this, the first principal component analysis spectrum, which we see on the left here, will show us, it will highlight all of the peaks that have about the same relaxation behavior. And the second component spectra will highlight those particular peaks that have a somewhat different evolution profile. So without any peak detection, without any analysis, we can quickly hone in on the parts of the spectra that are changing in this relaxation series. And that's just a general idea that we can use for any kind of spectral series, and it's very easy to do. Now I'd like to turn our attention to the idea of using this kind of principal component analysis in a quantitative way. And this is an aspect that is actually, it doesn't seem to be very well understood by folks who try to apply principal component analysis, so it's worth discussing. As we explained, we have some high dimensional space with objects that represent each spectra in the series, and we fit a given principal component as a high dimensional straight line through that data cloud, and that multidimensional straight line gives us the orientation for a projection, and we use that projection to make clusters and so on of the different groups. So this is how it would work for a pair of measurements on 2 different samples. So replicate measurements of one sample, replicate measures of another sample, and we can see 2 groups, and we can measure a similarity in terms of a distance between these clusters, between their centers or their closest members, whatever. But we can use this to generate an interclass distance for these 2 spectra. But what happens if we add a third group of spectra for a third sample? Okay, now if we add data for a third sample, of course, the relative positions of spectra for the original 2 groups doesn't change, right? These spectra are all the same, so their positions in this high dimensional space do not change when we add a third group of spectrum, but the principal component that describes this new group of 3 spectra will be different in orientation than if we only had 2 groups of spectra. And so for that reason, the direction of projection is going to be different. And so the original interclass distance that we saw in the first case when we only had 2 clusters is going to appear differently when we add a third. And so for this reason, these PCA results can only be used quantitatively when we compare 2 samples at a time. Okay, so that is one of the caveats that we're going to do. And to understand how this works better, we're going to review an alternate way to consider principal component analysis. And this way, I think, actually makes more sense to a spectroscopist. We can also think of principal component analysis as a matrix decomposition method. And to explain how this works, we're going to use a collection of synthetic data that's along the lines of what we saw. We will say that we have 2 different samples, sample A and sample B, and in each case we measure a collection of replicate spectra of these 2 samples. Now, in this synthetic case that we built here, sample A has 2 peaks, sample B has 2 peaks. The bigger peak in these 2 samples is the same, but the smaller peak in these 2 samples slightly displaced, and one is a little bit broader than the other. So if we looked at them in overlay, they would look like this. So 2 samples, here's what they look like in overlay, and here is what the spectral series looks like if we kind of spread it out in 3 dimensions. Now, if the samples themselves don't change during measurement, and there's no systematic drift during the measurement, we would expect that there is no systematic variation between the spectral measurements within a class. And so we take this collection of data where we assume there is no substantial systematic variation within a measurement, and we use the algorithm called the NIPALS algorithm for calculating one principal component. Okay. The principal component will consist of a collection of scaling values that we call the scores and a loading spectrum. The loading spectrum is a linear combination of all the spectra in our input series. And the score values are the coefficients that we use to multiply the spectra to generate this linear combination. And these score values are chosen so that the synthetic spectrum that we generate by linear combination describes the complete set of data as closely as possible in a least square sense. So we're combining all of these spectra into a single spectrum that's the best approximation of the series. And in practice, when we do that for data like this, the first component spectrum that we get looks very much like an average spectrum of the spectral series. So that's our first component. We can take that first component, which is a set of score values and a loading spectrum, and we can multiply these together and subtract them from the original spectral data, and that will generate a residual. The residual can then be used to calculate a second component, and we will use exactly the same procedure. We will find a set of coefficients that we can use to combine all of the spectra in this residual to generate a loading spectrum. And we generate the loading spectrum in order to describe this residual series as closely as possible in a least square sense. And when we do that for a case like this, which has 2 sample classes, the loading spectrum will look very much like a different spectrum of the original data. And if there is no substantial systematic variation between the 2 samples, within each sample, 2 components should describe everything in the spectral series because there are only 2 samples and 2 spectra should be able to represent them. But in fact, we can repeat this calculation as many times as we want to generate additional components. And if we see any substantial result in the third component, then we know some kind of systematic variation occurred during the measurement, either a change in the sample during the measurement or a change in the measurement details and conditions. So here's a test case for this method to illustrate it. One of the clinically relevant changes that can happen to these biopharmaceuticals is oxidation of methionine residues on the surface. So this kind of post-translational modification is an important detail to keep track of. And as a test case to demonstrate the sensitivity of this method, we prepared versions of our reference monoclonal antibody that's we call the NISMAB. We prepared two versions of this, each oxidized in different ways. In one sample, we simply dumped the antibody in with peroxide and oxidized it directly. In another sample, we first bound the monoclonal antibody to a protein A column, and protein A binding with the monoclonal antibody will protect particular methionine groups on the antibody surface. So these these particular methionines will not be oxidized as readily as the other ones. And so we prepared two samples like that. Now, it turns out that in the particular way that we prepared these samples, both of them are almost completely oxidized. Both of them are almost completely oxidized. So the methionine signals that would normally be there are almost completely gone. And what you can see in a detail here, in gray are the methionine signals that you would normally see in the native antibody. And in blue are the signals from the antibody that was oxidized directly. And in red are the signals from the antibody that was oxidized on the protein A column. But the contour levels for these two spectra are drawn at 5 times lower levels than what we see for the native spectra. So these residual methionine signals are very tiny, and there is only a little bit of a difference between the two. So very hard to tell the difference between these spectra by eye, and there is very little chemical difference between these two samples, just a slight difference in oxidation patterns. But can we tell a difference between these using this principal component analysis method? Well, what we did is take versions of these spectra and change them and distort them in different ways, prepare them in different ways, pre-treat them in different ways to figure out whether or not and under what conditions we can tell the difference between these two sample classes that we know are different. And so what you see in this graph are different cases where we added different amounts of additional experimental random noise and judged the degree to which we could make a successful classification. So on the horizontal axis is the amount of additional noise that we added to the data relative to its existing noise level. And on the vertical scale is how easy it is to tell the difference between the two sample classes. So the higher up on this axis, the higher and easier it is to make a good classification. And this gray band is the confidence limit that goes from a 95% to a 99% confidence limit. So anything in this gray band or up is a good classification. And what we see is that, first of all, we can change the noise level by a factor of 2 or 3 in this data and still get a good classification. So that's one thing. And then we experimented with all different methods of pre-treatment. For example, whether or not we could bin the data, apply smoothing, the effects of phase distortion, on how many increments we measure in the indirect dimension. And in almost every case, we can still get good separations. But interestingly, the thing that always ruined our ability to make a good separation are systematic baseline distortions in the data. And so what we saw in this case is that if we add a synthetic baseline distortion of about 0.24% RMS, we can no longer make a good classification. And this RMS is about half the random noise level. So this is only a small systematic distortion that you wouldn't even see in the spectrum by eye, but it's enough to damage these results. And so what that tells us is, on the one hand, the method is very tolerant of random noise, but on the other, even if you measure spectra with low signal to noise, you still have to use good measurement technique and good pulse sequences, because you have to have spectra that are free of systematic artifacts. Now, another interesting thing is that, as we've seen in our other examples, the principal component analysis results can be visualized as spectra. And so what we see here in gray is the principal component analysis spectrum 1, which is like an average spectrum. So we could think of this as, in gray, as what the native spectrum would look like. And in red and blue, you see the parts of the spectra that are different between these two partially oxidized samples. And so what that tells us, which is interesting, is that the changes to the spectrum are not just limited to the methionine region, they are distributed throughout the spectral data. So the fingerprint information is all over the spectrum. It's not just in one region, which is very good and useful, because it means that no matter which analysis region that we choose, we can still get separation. So I can get good separation if we use this big gray area, still get good separation if we use this pink area that doesn't even include the largest spectral signals or the methionine region. So that's an interesting aspect, that in many of these cases, the information that we care about is distributed throughout the spectrum. And what that means is that we can potentially perform this measurement in a way that does not result in data that you could analyze visually, but still results in data that you can make a useful classification. And in fact, if you think about this, when we Fourier transform time domain data, we don't add any new information. The information in a spectrum is the same as the information in an interferogram. So we can apply this principal component analysis technique to interferograms. We can apply it to regular Fourier transforms of non-uniformly sampled data. All of these different circumstances will yield about the same results, almost regardless of whether or not we have time domain data, frequency domain data, regular Fourier reconstructions, or full-blown Nuss reconstructions. So that's also interesting too. So that is my overview. I hope I've convinced you that PCA is an excellent way to deal with any spectral series. I hope you understand the mechanisms of PCA, and you're convinced that we can apply it quantitatively. And in particular, that it can be robust with respect to random noise. It can permit us to make measurements for classification purposes that can't be analyzed visually, but can nevertheless be exploited. So that's my overview. Thanks for your patience. Looking forward to your comments.
Thanks, Frank, for a fantastic, beautiful, insightful presentation. As always, very clear. I'm sure that some grad students and others wrestling with spectral analysis have questions that they haven't posted yet, but let me start with one that I've got myself sort of from past experience. If you're doing, say, a pH titration where different residues have different pKs.
Yeah.
Now, not everybody shifts at the same time.
Right.
Can one facilitate the analysis with those very crowded spectra that cross over and all kinds of things?
Yeah, absolutely. And folks actually use PCA for this purpose all the time. And there is a really good software package called Trend that is actually specifically already arranged to take nonlinear curves in principal component analysis space and characterize them. So you can put in almost any kind of data that you can imagine, including photographs, and get evolution information extracted this way. And I've seen specific cases where folks prepared a pH titration, and the pH titration went the way they expected in terms of pH, but it changed the ionic strength of different samples. So when you actually look at the pH curves with principal component analysis, there are discontinuities that you were not expected to begin with. So it's actually a really quick and easy way to reveal details like that in the spectra without the need to extract individual evolutions.
Thanks.
You don't have to see the individual peaks.
Right, right.
Ed, can I ask 2 questions?
Yeah.
One is, you do this by, you said, by doing least squares minimization. And I'm wondering, why that rather than some other norm, since these days one can minimize basically anything. And a related question is, at the end, you said basically it doesn't matter, how you input the data, whether it's time domain or frequency domain.
Right.
But in other applications of PCA, for example, if you're doing PCA in a structure sense, it can matter whether you're using Cartesian coordinates, or dihedral angle coordinates. So
Right.
why in this case is it so independent?
The reason it's independent is because when you do a Fourier transform, you replace one orthogonal basis set, which is a bunch of sine waves, with a different orthogonal basis set, which is delta functions at different frequency positions. And as we know from Parseval's theorem, the magnitude of time domain data is the same as the magnitude of frequency domain data. So the objects that represent these two things in principal component analysis space have the same magnitude. And since they both are orthogonal to each other, they just correspond to rotation in the principal component analysis space. So in other words, the cloud that represents time domain data in PCA is the same as the cloud that represents the corresponding frequency domain data. It's just rotated into a different position. So that's why. And that is not the case whether we consider, say, a protein structure in terms of Cartesian coordinates compared to a protein structure in terms of dihedral coordinates. Those are very different representations of the same data. So it's not the same kind of comparison at all. Does that make sense?
Oh, sounds like I better reread a section of my own book about that.
A follow-up or a question that actually preceded the point raised by Art, by Victor Beaumont. He says, great talk, but what advantage is there in performing the PCA on the interferogram?
Honestly, there isn't a super example of performing it on the interferogram. And in practice, it turns out to be better to perform it on the spectrum because it's hard to properly normalize an interferogram. And it's also hard to perform good phase correction without looking at a spectrum, even if it's not a very good spectrum. So in practice, all of the best results that from our hands can be computed from spectra. But one of the reasons why you might consider using interferogram data is that you don't want to use a nonlinear reconstruction for the data that might change relative intensities. But it turns out, so for non-uniformly sampled data, it turns out that if that's how you feel about a nonlinear reconstruction, you can just use an ordinary Fourier transform. All the original information that was in the measured experimental data is still in the result. So it gives you a result like the one we saw in this slide. This slide is generated by a regular Fourier transform of non-uniformly sampled data. but it still contains all the same information. So in a sense, when we perform a NUS reconstruction, we're not adding much information to the data. All we're doing is reorganizing it in a form that can be analyzed visually. When we use PCA, we don't need that anymore. So we can work with data like this. The reason why that's interesting for us is that we can generate spectra from measurements that might have only 4 or 5 increments in the indirect dimension that would never really generate a meaningful spectrum, but they still have enough information to make a classification like this. So that's my long-winded answer. Does it make sense?
No, makes sense, Frank. But one follow-up question then is, you highlighted the problem with baseline distortion.
Yes.
It's very difficult to do baseline correction on data that have only been Fourier transformed.
That is absolutely true. Yeah, right. So for that reason, as I mentioned, it's even fair to say the Achilles heel of this method is measurement technique is super critical. You have to do everything that you can to make sure the data are free of systematic distortion. So that means very carefully set up initial delays and all the other things that you would do. Now, for us, that's not bad because we expect to take the same pulse sequence and parameters and employ it in the most consistent and reproducible way possible to make measurements like this. So.
Sure. Hey, and another question by a dear friend of many of us, Jim Prestegard. He asks, can PCA be used to tell how many states are involved in a protein observation, like ligand binding one site or 2 sites?
So there's no limit to how complex the organization of the data is. You could have as many sample classes as you want, and PCA is not any harder to do with a large number of spectra as a small number of spectra. It's the specific case where you want to measure a spectral, measure quantitatively a difference between 2 groups of spectra that you have to consider how many other kind of sample classes that there are.
All right. Another question. It was a nice explanation and very informative. I assume in your case, protein might be well behaving. Now, if there is a multi-domain protein, and let's assume it has intermediate exchange, in that case, how does PCA classification look like when you compare 500 and 700 megahertz?
That is a good example, and it depends on the nature of the spectral difference, right? So in the case that we saw before, like, let's see, where is that one? So we saw this, okay? So in this collection of data, the 2 samples have about 450 residues each, but in one of the samples, there is an additional 4 residues on the N-terminus of the 2 chains of the protein. So it's a minor difference, but that one is enough to allow us to tell the difference between these 2 samples in a collection of spectra between 500 megahertz and 900 megahertz. So this is, to me, this was remarkable and unexpected. I didn't really think that we could so easily tell the difference when comparing a spectrum at 500 megahertz directly to a spectrum at 900 megahertz. But it depends on the degree of difference. If we wanted to do this classification on, say, 2 samples that had the exact same sequence, but only differed in oxidation pattern, like our test case, we probably would not be able to discriminate based on measurements mixing measurements at 500 megahertz and 900 megahertz. So we have a different way to account for the expected range of measurement variation, and it just depends on the circumstances. So if you intend to compare spectra that are measured on different instruments. We use a collection of spectra that represents the expected measurement variations between these two instruments. And that's how we deal with this problem.
But I think we should move on to the second
Yes.
speaker of today.
Thank you so much, everyone.
Thank you, friend. Tony, it's yours.
Thanks, Marcus, and welcome to everyone. Thank you very much for asking me to introduce another one of the speakers in this webinar series. Delighted to do that. It's such a nice group to interact with. So Clemens Glaubitz joined my lab in 1993 as a diploma Arbeit project student from Leipzig, which is where Klaus Garvisch and Klaus Arnold were. He then must have liked the place because he stayed on to do graduate work as a DPhil student between 94 and 98. And he was elected to a Rhodes Scholarship during that time. This is a very elite and prestigious group. Only 33 Rhodes Scholars who are non-Americans get elected each year. And he joins the ranks of people like Lord Florey and James Fulbright, Edwin Hutton, for example, and Bill Clinton, of course, as Rhodes Scholars at Oxford. And then he's stayed on for a postdoc between 1998 and 2000. So he has a talent which, of course, people recognize within this group, not only tackling the technology and the methodology. I think he's well known for the magic angle sample spinning method, where these powder patterns that were a real pain in solid-state wide-line NMR could be resolved into very high-resolution narrow-line spectra by spinning a sample at the magic angle. And most of these samples, of course, membrane samples. So the long-axis rotation of the molecules themselves averaged out all the dipolar couplings. And this is actually a proton spectrum here of a natural membrane. He also had a good eye for biological problems, which is also important. He worked on a wide range of systems, not least bacteriodopsin, for which he was well known from the Mayos, and then rhodopsin work, resolving the structure of the retinal prosthetic group in the protein before the protein crystal structure came out, and showing that the retinal was significantly distorted, something that wasn't known before. Worked with myosin, he's worked with M13 coat protein, and also with smaller other proteins and peptides. And he continued that project as he went on to Germany. He did the thing that most students do. He drank cheap beer in Oxford. He went punting in Oxford, many people do. Here he had the privilege of being punted by Paul Johnson, who what many of you will know. And he was with Gerhard Grosvenor, with whom he had a long-term friendship when they were in Oxford. These are two photographs that I found quite easily from my compilation of lab photographs, but I couldn't find any more serious ones of him in the lab. But he was clearly very enthusiastic joining in the Christmas parties. And I don't have a picture of his PhD examination, his DPhil examination, except here he's fighting with a champagne bottle to try and get the top off. And he was examined by Peter Hoare in Oxford and Standard Peller's examiners. He spent a year or so with Malcolm Levitt in Stockholm before Malcolm came back. He secured one of these prestigious eminent group leader positions, and then he has been in Frankfurt, as people know, since then. We had an interesting conversation before his postdoc about whether he should go and join Ian Campbell and do solution state NMR or stay with solid state NMR. I'm delighted he stayed with solid state NMR, and today he's going to talk about NMR detection of light-dependent processes in membranes. So over to you, Clemens.
Okay, thank you very much, Tony, for the very kind introduction and memories of my PhD time in Oxford. So I think, yeah, I think this should work. You should be able also to see it. So my talk will be dedicated to solid state NMR studies on light-dependent processes in membranes. So in my lab in Frankfurt, we mainly work on membrane proteins and membrane protein interactions and things like that, covering GPCRs, transporters, receptors in general. So, but today I will restrict myself really to two topics which are really connected to the question of light control or optical control in the context of membranes. So the first topic will address the use in the background of photo cages and switches as photochemical tools in the membrane. Now, these things are usually used in the context of in vivo in situ studies, in cell biology, for example, but very often on an empirical basis, and very often the photochemistry on the mode of action of these systems is not so much yet understood in great detail. So that's something we are going to look at into that. And the second topic, I will briefly show you some examples on our work on photoreceptors, which is another class of light controlled systems which are native. The first group is, of course, a artificial way of putting light control into our system. So the method of choice is, of course, still solid state NMR, as Tony mentioned. I selected that already for my PhD times and stick with it since then, because it's, of course, a very useful and extremely powerful method for studies for everything around biological membranes. And we use two flavors of this technique. Everything is based on magic angle sample spinning, but we use a lot, of course, high field, multidimensional solid state NMR, non-frozen samples, uniformly labeled samples for getting assignments and this type of things. But at the same time, we also do make a lot of use of dynamic nuclear polarization, which better works at lower fields and lower temperatures. But this is, as you will see, a fantastic tool, especially in the context of investigating photoreceptors. So now let's get into the first topic, light control of membrane proteins, and what's kind of the motivation behind that? There are two motivations. First is if you have a membrane protein or a cellular membrane, it would be nice to have a disturbance-free way of controlling all kinds of biochemical reactions which might be ongoing in this system. The first motivation. Second motivation is it might help to do this under in vitro conditions to understand really these tools and the mechanism, mode of actions of these tools in greater detail. And the third reason is there's really a practical reason. So you can see here on the left, if we do magic angle sample spinning, we do work under sealed conditions. The MRS rotor is a sealed system. It's pretty difficult to do a sort of a titration experiment. There's a great advantage if you compare this to, for example, the solution state NMR. There you have not only very often nice resolution, but also you have a system where you can more conveniently put stuff in during the experiment and do some sort of titration experiments. So there are lots of reasons to think about that, whether these type of experiments could be carried out under magic angle sample conditions. And the system I'm going to use for these studies, for this demonstration, is a little enzyme, deacylglycerol kinase from E. coli. It's basically phosphorylating DAG, deacylglycerol, at the expense of ATP into phosphatidic acid. Yeah, so it's a very important system in the lipid biosynthesis of gram-negative bacteria. And it's also on top the smallest known kinase. It's a homotrimer. So the nucleotide binding sites are here, the lipid binding sites are here. And the structure is known and it behaves pretty well. It seems to be kind of a good system to do this, to carry out these kind of studies. But the aim, what I'm going to demonstrate is the following. First thing we will check out whether it's possible to trigger and release ATP by an uncaging reaction during an MRS experiment. So this would bring one part of this enzymatic reaction under light control. The second would be to release by light or uncage also the lipid substrate in the membrane. This is more challenging, but this would be the second branch of this enzymatic reaction. Put that under light control. The third part would be to see whether we can actually modulate the membrane properties by light by using, for example, photoswitchable lipids, to see whether we can modulate in that way protein properties or maybe functional properties of that protein by light during the NMR experiment. So the protein itself behaves nicely, as I said, and offers very good starting conditions. So we were able to completely assign the trimer in the lipid bilayer in the upper state with bound nucleotide, with nucleotide analogs, and also with bound lipid substrates. We were able to observe titrations and look at chemical shift perturbations and so on. And under all of these conditions, the trimer is symmetric in contrast to the X-ray structure, which shows some sort of asymmetry, which is probably based from crystallization contacts. And spectra nice. So with the full assignment, we can basically go ahead and then look at the response of this protein under light controlled conditions. The second important aspect I mentioned, this is a trimer, fairly stable trimer. And here this is a use for DNP spectroscopy. We try to look at specific interactions at the trimer interface in order to understand what keeps the trimer so stable. And this is something, one way for checking that out would be, for example, by type of TDOR experiment where you do segmental labeling. You label one subunit with carbon 13, the other with 15N, and basically you disrupt the trimer, reassemble it. Of course, you have to check the functionality, and then using the TDOR experiments, it will show you if cross peaks come up, then these will be basically cross peaks across the interface. And this is a control experiment here, fully carbon labeled. As you see, only natural abundance nitrogen to carbon. But if you have a mixed labeled complex, you do see additional peaks coming up, like this one here. This is really a spectroscopic representation of a salt bridge, and we were able to identify these salt bridges. And there's exactly 3 salt bridges which keep this trimer completely stable. So this is the starting condition, and then Julian in my lab then started to work out a procedure for triggering the reaction of that protein, observing this reaction of that protein under MRS NMR conditions by light. So for that, he replaced basically if this is done under the right conditions, and if the probe head is modified in the right way with a fiber here, as it said, illumination can take place under MRS NMR conditions, it is possible to release basically the ATP and then to see how it reaches the protein, and then the reaction in the liposome starts and takes place. Yeah, this is something we can see here. This is this is the phosphorus MRS NMR spectrum of our sample. These are the lipids. Here's the NPA ATP. We flash by light. It gets released into ATP, and then after time, the reaction starts and it turns all into ADP. Now, here are the progress curves shown on the left here. And this shows you can, this part of this type of branch of this enzymatic reaction can be put under light control if the conditions are right. So this took quite a time to find the right experimental conditions. Now, how about the lipid substrate? This had to be especially synthesized by Alex Hecker, as my collaboration partner here in-house in the chemical biology department. And they managed to prepare a diacylglycerol with a photochemical, with a cleavable group here. This has been reconstituted into the liposome together with the other lipids in the protein. And in the MRS NMR rotor, we were then able to cleave basically this lipid, activate the lipid, then observe basically how this lipid is being phosphorylated. And here in this case, we had to use here gamma ATP, the ATP gamma S, in order to have a nice chemical shift change here of the phosphorylated lipid product. But you can nicely see here that the lipid product here comes up. So this is also possible in the membrane, basically to release the lipid substrate by light under MRS NMR conditions, which is quite nice. And again, you do see here the progress curves on the right, from which kinetic parameters could be obtained. Now let's come to the membrane. What would happen if you try to change the membrane properties by light? Now, photoswitchable lipids have been used quite a bit in cell biology to control, for example, liposomes, curvature, or control ion channels, receptors, or enzymes, or nuclear receptors. And there's a kind of a large library of various photoswitchable lipids available. But so far, how they in detail actually interact with the bulk lipids and how this causes disorder or order in the membrane, how this feeds back out the proteins, this hasn't been studied so far. So the lipids we have chosen for these studies are lipids which carry here azobenzene group in one of these chains here. And for that, we teamed up with the group of Dirk Trauner in New York, who is an expert really on putting almost everything under light control using these azobenzene groups. And they prepared quite a lot of these photo-switchable lipids for us. And Mahmoud in my lab, my PhD student, worked out basically an optimized liposome mixture for investigating the effect of these photo switching onto the membrane properties. Yeah, so we have chosen POPE, POPG. So these are the main components, lipid components of E. coli membranes, the inner E. coli membranes, where also the protein, the DGKA, is residing. And we had some basically liposomes which contain these lipids in this ratio, plus this membrane protein. And upon light illumination in the UV range, AZO-PC switches from the trans to the cis state, and then thermally relaxes back over a time period of 65 hours, something like that. So the first task was then, of course, to see, can we indeed reach a sufficient photo conversion? And this is something one can nicely observe by proton MIS NMR, because it's just looking here at the azo switch in the trans state. We have here these proton signals here of these rings, and they switch into cis their position. By that you can see that we have here pretty good conversion rate for our photolipids. Yeah, and it's the case that after flashing light, we have a conversion, and then it decays over 65 hours back basically to the trans state. So the first question was then to see how in that proteoliposomes, how the photolipid interacts with POPE and POPG. For that, we did a simple experiment, basically using a nosy MIS NMR experiment, by which we could observe NOE cross peaks between the photolipid and the bulk lipid. Shown is here just the POPE. What we see is the trans state, trans AZO-PC, and so you can see this is extended. So we do see here kind of quite some interactions along the chain. I've just picked out these two. We can illuminate the sample, switch to the cis state. We do see here that the interaction pattern changes, of course, and also we only some of these rings here interact with some of these lipids. Yeah, but this is also remarkable that methyl group interactions still take place. You do see interactions between the methyl groups and some of the ring protons here. Because the switching apparently must create some sort of a void in our membrane. This must be filled, of course, thermodynamically by the membrane, and our hypothesis was, well, this must create some sort of increased disorder here in the membrane, which must alter then the membrane lateral pressure, for example, on the membrane phase behavior. And in order to look at that in some more detail, we were characterizing basically the membrane by order parameter measurements. This can be done by, this is a classical way of separated local field experiments. There are many versions available. So this is here dipolar order parameter measurements using this R1817 pulse sequence described in this paper. As you can see here in the dark and in the light, the patterns, the dipolar patterns along the chain do change significantly. Especially this one here, the largest one, this is an overlap, superposition of these grayed out carbon groups here, does show some significant changes localized. The others are less affected, but these ones are significantly affected. If you have a look here, what's going to happen? We had a closer look. We deconvoluted that spectral region. Again, this is in the dark, illumination and relaxation back to the dark. You do see here basically a nice dipolar pattern with these subspectra corresponding to all these carbon groups here. You illuminate, then you see some more narrow patterns come up. So some get more disordered, have a much smaller order parameter, and then after time it relaxes back. So if one systematically analyzes that for every carbon position, we do get order parameter profile. This looks complicated at first glance, but it's not so bad if one looks a bit closer, because the largest change really occur here in the middle of this chain region, which coincides basically with the region where we saw also from the NOSI experiments the NOEs between the azo group and the protons here in the lipid chains. And we do have significant but highly localized changes in the order parameter. So the top order parameter profile, what you see here, is basically the trans state in the dark. Upon light, it switches here into the into the cis state. And you see for some of these groups, CH2 groups, significant reduction in the local order. Yeah, so if one calculates an average order parameter for that region, changes reduced from 0.22 to 0.16, which is really remarkable. Yeah, so the switching of the lipids has really a highly localized effect onto the order of the bulk lipids. Question is now how does it affect the protein itself? And for that we recorded some simple NCA spectra. So we do have the assignment, as I mentioned, we do have a good resolution. And for that, we could basically then record the spectra fast enough directly after illumination, and then later on, whether we do see any chemical shift perturbations or intensity changes in our spectra. Now, interestingly, we did not observe any significant chemical shift perturbations, but really substantial changes in the cross-peak intensities. And in principle, after light, many of the cross-peaks became smaller, but not randomly, but really localized in certain areas. For example, here on the surface helix, we have certain spots which become really disordered, highly flexible, not disordered, I should not say, but highly flexible. And we do see really a substantial response of the protein dynamics, at least in a qualitative sense, with respect to the increased disorder, localized disorder in the membrane. Yeah, and this is something that the protein responds in such a way, or that, for example, the surface helix here responds in such a way. This is an initial hypothesis we always had, because based on the function of the protein, it's supposed to respond to osmotic stress. Osmotic stress does alter membrane properties, a lot of pressure in the membrane, and the surface helix here is supposed to play a role in that. And so far, we were not so surprised to see significant changes taking place here. Yeah, and on the right, you see here on the X-ray structure plotted the areas where you have the largest changes in increased molecular dynamics. So the second question is then, okay, this protein seems to get more flexible. So these NCA experiments, I should say, involves two cross-polarization steps based on dipolar couplings, and therefore it's basically, the intensity change takes place, it reports on increased dynamics because dipolar couplings are pre-averaged. How would that now affect basically the function of the protein? And for that, again, we recorded real-time phosphorus NMR data on the protein, as we have shown before, as you have seen before, and looked basically in the trans state of the lipid and in the cis state of the photolipids, whether any change in the activity of the protein takes place. But just to explain the progress curve here, again, we see here the consumption of ATP, we see here the production of ADP, and initially we see here the lipid mixture, ACPC, POPG, POP. At the end of the time, we see also phosphatidic acid coming up as a lipid product. If you compare this experiment, the trans state and the cis state, this is an interesting difference because you see the cis state upon illumination runs a lot slower. Yeah, here we have completed the reaction already, while here it's almost twice as slow under illumination conditions. So these data, why could that be? Well, some more data will be needed for that, but one explanation could be, I mean, we have two binding sites, and there has to be some coupling between the binding sites. We have the lipid binding site, we have the nucleotide binding sites. If the protein starts to get more flexible, maybe we lose allostery between these binding sites, and this might affect the activity of that protein. So basically, what we, in technical terms, what we could show is we, under MIS and our conditions, we were able to show we can bring the ATP part of the reaction under light control. We can release the lipid substrate, and we can manipulate the membrane itself by light. And most importantly, this ACPC causes really a highly localized disorder in the membrane, and this feeds back onto the protein structure itself, or not on the structure, but on the protein dynamics, and in a highly specific manner, and also affects its activity. Now for the second part, I would like just to switch then really to real photoreceptors, which are natively under light control, basically. And this is something, an interesting story, because as Tony mentioned, during my PhD, I did already some work on rhodopsins. And when I started my independent career back in Germany, I thought I'd stay away from that, because it looked at that time, late 1990s, this is overdone, this field. Most of it has been done. Most people considered rhodopsins or photoreceptors as overstudied. Even that at that time it wasn't true, but this is true. A huge amount of work was done, and we did a lot of focus on other proteins. But in the past 10 years or 15 years, there had been a revolution in the field, because thousands of new microbial rhodopsins were discovered in the context of biodiversity screens, genetic biodiversity screens, and basically they're found everywhere in all branches of life, extremely abundant. For example, this one, proteorhodopsin, is a light-driven proton pump. This is actually the most abundant photoreceptor on Earth known by now. This pumps protons. You even find sodium pumps, which is very surprising that you find something like that, because you have a heavy sodium has to cross basically the charged shift base in the middle of the protein, or you even have light-controlled channels like channel rhodopsin 2, which is the founding cornerstone of the field of optogenetics. And what's amazing in this field, and this becomes more, these questions come up more and more, is the more and more 3D structures we get, how these apparently similar structures and architectures can allow such a huge diversity of functions. So this is really, really interesting, and this requires really investigation of these proteins at very fine details. And NMR is here in a very good position to contribute some important insights to that. And this is just a cartoon just to illustrate what we are interested in, because what we want to do is, of course, we look at these proteins ideally in different photointermediate states, trap them, look at chemical shift changes, and interpret them in terms of 3D structures, link them to optical data, ideally by QMMM methods, in order to get a complete picture how these proteins work. So we teamed up with, for example, the group of Xiao He in Shanghai. It's really a world expert on fragmented quantum mechanical calculations on large biomolecular systems. So he's able to really nicely reproduce experimentally and theoretically our measured chemical shift CSA tensors, for example, on rhodopsins in different retinal proteins. But I don't want to get into detail with that. I just want to pick out 2 examples in order to illustrate really what kind of insight one can really uniquely contribute from the type of NMR spectroscopy which we do. And all these proteins go run through a photocycle. So we need to look into these photocycles and photocycle intermediates. And of course, with NMR, this is pretty tricky to do this in a time-resolved way. So we need to trap these intermediates or stabilize them. For that, this is done thermodynamically by mixture of illumination and freeze quenching, fast freeze quenching, or illumination at low temperature, and then just trapping first intermediates depending on how the energy landscape here looks like. So this in principle can be done, this works, but this goes with the fact that you need samples which are transparent for light. You end up with very thin layers of liposomal samples. This reduces the amount of sample you can use a lot. You can use basically, and you need low temperatures as well. So fortunately now with DMP, we have a tool at hand which is almost ideally suited for these type of studies, because DMP, cross-effect DMP, works beautifully at low temperatures. We can equip our system with light sources and feed in during the experiment not only our microwave, but only basically laser or LED light in order to trap certain intermediate states. And based on that, we can basically then dissect certain interactions and look at events in the photocycle, which one would easily miss by any other method. Yeah? And one of those examples I would like to show you here. So one observation which came up in the last years was the fact that many of these rhodopsins form big oligomeric complexes, pentamers, for example. many pentamers. Here is a sort of a phylogenetic tree of these microbial rhodopsin family. In the archaeas, you have many trimers, and in the bacteria, higher organisms, yeasts, and so on, you have algae. Here, very often you find pentamers. The question is always: is this just a way of best packing of these proteins? Or is there any function to that? So we looked into that and tried to discover whether there are any specific interactions first occur in between the interface in these pentamers, and also whether these interactions take part in the photocycle. Yeah, and I show you one example here. So in this pentamer of proteorhodopsin, this proton pump, there's one histidine here located here. We had to look at this histidine because this makes a contact within one protomer with an aspartate. This is the primary proton acceptor of the proteorhodopsin. And it seems in close proximity to tryptophan across the interface here. And this looks like a sort of a triplet, a triad. And we try to see whether first, what's the histidine doing during the photocycle, and whether this has any functional meaning. And this is a great example for DNP, because if you look at this histidine in the ground state, in the dark state, we see the spectra here of the histidine, and it's really basically primarily here in the tau state. Yeah, as you see, you can see this here nicely from the chemical shifts. Here's the protonated shift base and where the retinal is linked to. If you illuminate the sample and trap this basically in the M state, the shift base is deprotonated, so the signal here disappears, as you can see. The signal changes. Yeah, you see it switches completely to the pi state, so it changes its tautomeric state, which was very surprising. And we were also able by measuring a data coupling between the nitrogens here by spin diffusion experiment, that the ring also flips. Yeah, so this is really a photocycle event at the interface, which wasn't known so far, because in most cases one would look around the chromophore and the known canonical ion pathways. And this was something really off target at the interface, something which really takes place in the photocycle. And does it matter functionally, or is it just an interesting coincidence? Well, it does, because as I said, it makes a contact here with the tryptophan. This also we measured here with these mixed label complexes by through-space TILO spectroscopy. And this is a transport assay in the wild type. It pumps protons. If you replace, for example, the tryptophan here, disturb this complex, these three amino acids, it pumps the protons in the opposite direction. If you replace the histidine, the photocycle runs a lot faster. If you replace the aspartate, the photocycle completely dies. So this seems to be like a pH-sensitive regulator. This is our hypothesis at the moment, which it sits at the interface and might help to adjust the activity of the protein to altering environmental conditions. So this seems to be a really new structural or functional motive in this type of proteins. So the last example, briefly, I would like to show you is another case which was also recently published. This addresses channel rhodopsin. So as I said, this is the cornerstone of optogenetics, and it's a light-gated ion channel, and it's great in cell biology. If you have a nerve cell, you express this in a nerve cell, and you use light, and you could sensitize or desensitize cells without using an electrode. So this is used a lot, papers going up a lot on the usage of channel rhodopsin, but the biophysical fundamental understanding of channel rhodopsin lags behind, because there's really a lot of contradicting data and open questions. Just to pick out one, it's just simply the photocycle is a lot more complex than what we had seen before. But there are two models I just show you, and one model, for example, shows you have here the ground state, light is absorbed, the retina isomerizes from I-trans to 13 cis, and the protein runs here through the whole photocycle. And here this state, this is very important, this is the desensitized state, which is linked to the channel open and closing, and this would come late in the photocycle. This is one model. The other model suggests that upon light illumination, you have branched photocycle, you have this one, plus the channel desensitization takes place immediately. So this forms immediately here. So this is the first contradiction. The second contradiction is in this state, what configuration is retinal in Al-trans 15 anti or 13 cis 15 syn. This matters because this will determine how long, or might explain the long lifetime of that state. Because if it's in this state here, it might take longer to relax back compared to this state. So these are questions. This has been approached by optical spectroscopy, Raman spectroscopy, and so on. So all these contradicting data came up, but this can be actually nicely resolved by NMR spectroscopy. And again, we have by illumination and choosing the right temperatures, we can basically trap these certain intermediate states. And just the first question, whether this P4 state comes early in the photocycle or late in the photocycle, can be easily addressed by illuminating the sample at low temperature and see which states come up, or whether by thermal relaxation you can reach later states. This is something we can see here. This is the signal here for this P4 state. And you see upon illumination in the dark, upon illumination, the signal does not come up at low temperature. You have to raise the temperature in order to reach that later intermediate states. So that's something one can already exclude that takes place. That this photocycle intermediate occurs already right after illumination. The second question addresses basically the configuration of the retina. So the easiest way is, of course, measure here distance from here to here. And then you could, of course, nicely differentiate between the one state or the other. So Johanna, who did the studies here in my lab, figured out an experiment. This was published in the SR26. This is a symmetry-based double quantum filtered pulse sequence, which with CSA compensated allows to measure long distances. And in the model compound, you can nicely see this is the double quantum buildup curve. This corresponds here to 13 cis. This corresponds to Al-trans. So you can nicely differentiate between these states. And then if you trap basically the P4 state, we do see here double quantum buildup curve for P4. These are the other states. This is pretty clearly here, this configuration which we obtain. So basically by these type of methods, you can, with a well-tailored experiment, get to answer really, really significant questions, which are passionately debated in the field. So before I come to the end, just want to highlight one other recent study from our lab, which is completely out of the context I have explained so far, but we have, of course, also very close collaborations here in Frankfurt, one, for example, with my colleague Harald Schwalb in our NMR center. And he started a very fascinating project on Mason chain complexes, ribosomal Mason chain complexes, and he was interested in understanding how the non-anonymous codon usage affects protein folding. And during those studies, the question came up, is one able to study basically disulfide bond formation or cysteine oxidation in the exit terminal of the ribosome? This is not an easy task, but fortunately, with the help of DMP, with a lot of optimization, with Linda Schulte, his PhD student, and Jafar from my lab, it was possible to record really nice spectra, as you can see here, from Mason chain from the Mason chain in the ribosomal exit tunnel, and to get really an insight into this biochemistry of folding and cysteine oxidation in this channel. So with this, I come to the end. These are the acknowledgements. This is my group. Here, I mentioned all the people from whom I presented the work as our collaboration partners. And I thank you for your attention, and I'm happy to take questions.
So I will handle the questions, Clemens. Thank you for a very interesting and sophisticated talk to show how to bring light to magic angle spinning NMR to illuminate membranes as well as membrane-embedded photoreceptors. So yeah, there are a couple of questions coming. So let's see. So Jörg Matisik says, wonderful data, wonderful talk. Is there any hint that in the rhodopsin protein photocycles, the rigidity of the protein is changed? And does the mobility or the local dynamics change during the photocycles?
Yeah, this is a good question and not easy to answer, because so I did show only really a tiny fraction of the data we have. So with the DMP approach, we work, of course, under low temperatures, and it's pretty difficult to say something about dynamics. And there we look really focused on really just very highly specific interactions. However, we do a lot of studies on these proteins also on non-frozen samples with uniformly labeled samples, looking by 2D, 3D experiments, also with illumination, and then in order to create a steady state. And there we do see a lot of interesting phenomena which we don't fully understand yet. Of course, there is localized. There seem to be some changes taking place. Some signals disappear, especially around the retinal. Retinal disappears under continuous illumination. So there seems to be some exchange ongoing, chemical shift exchange, or line broadening. Many other parts of the protein remain relatively rigid. So this is not a protein. These proteins do not undergo huge conformational changes, but this is really subtle, small changes, really on strategically important regions. But this is something we are looking into it. But of course, DMP will not help in that case. For that case, one needs really high field NMR, uniformly labeling, and plus illumination, and then to see under continuous illumination whether we can extract some data from that.
So maybe let me follow up on that question. Since you have quite some of these, a lot of your samples emphasize site-specifically labeled, systems, even though these proteins are large. Have you ever seen signs that some sites are broadened more than others, so that you might get some information about dynamic disorder at high temperature based on a DMP temperature line?
Under DMP, yeah, so as I said, so under DMP, we almost only work on selectively labeled samples. At high field, non-frozen samples, we always work under uniformly labeled samples.
Right.
So the DJK, what I did show you, this is was not DMP. So we didn't look into that. But of course, I mean, residues on the surface of a protein are affected by more paramagnetic quenching. Residues which are more flexible broaden more, show more heterogeneous broadening compared to those buried inside of the protein, of course. So this type of information one can extract, of course.
So, yeah. So for example, for your, I think it's proteorhodopsin, where you're looking at the interface, right, between subunits.
Yeah.
You have histidine and tryptophan and so on. Have you seen whether these signals are always similarly broad, or maybe some sites are well-defined, structurally robust? Just curious.
They are well defined. I mean, I would say. I mean, if in some cases we do see broadening. The lines are so broad that this is not really. It's really not coming from the paramagnetic broadening. It's really heterogeneous broadening because of the flexibility that you almost sample to full conformational space. Or you must sample the full conformational space. You see like line broadening of 10 ppm or something like that, which is really remarkable. Yeah, but with the histidine, I mean, the lines were broad. I mean, they're a lot broader than in non-frozen conditions, but they were always reproducible. They're always the same if you mean that. Yeah, so it's
okay.
Yeah.
So,
could I ask a question?
Sure. Yeah.
Well, I was also wondering about the histidine related questions. I didn't follow completely how you assigned the tautomeric states. You said something about DAR, I think. And related, did you also measure the pKa of histidine?
So the tautomeric state is this is uniquely linked to the nitrogen and to the carbon chemical shifts. That's something you can read out from the chemical shifts. There are some specific change of one or two nuclei in there. From that you can identify that. And
mainly based on the nitrogen shifts.
Nitrogen shift, yeah, and there's one the C gamma or one of the C delta 2, I think. This has been empirically shown that it's really a very sensitive reporter. So this is easy. And the other was the ring orientation. This was not directly linked to the tautomeric state, but the ring itself flips. Yeah, and there we measured basically, this was just a PDSD experiment, nitrogen PDSD with a very long mixing time, nitrogen. And in one case you do see cross peak, in the others you did not see a cross peak, and this means that the ring must have flipped.
Okay, thanks.
Yeah.
Okay, so I guess given that it's half an hour past the hour, we can switch to the informal session.